Welcome to Genomics Now, a podcast series where you can learn how genomics is developing in England's NHS. This podcast series is recorded in 2024 and is part of the North Thames Genomic Medicine Services Educational Toolkit. In each bite-sized episode, we explore what is changing in genomics. We answer the questions on genomics we've been asked by the different healthcare professionals. We talk about the ways genomics may impact different healthcare roles, and how genomics can be used to successfully improve patient care. Hello, I am Dr. Shazia Mahamdallie, and I'm part of the North Thames Genomic Medicine Services Workforce Education and Training team. In today's episode, we welcome back Dharmisha Chauhan, the lead pharmacist in the North Thames Genomic Medicine Service Alliance. Dharmisha, hello.
Hi, thank you for having me back.
No problem. Dharmisha, let's jump straight into today's episode, where you're going to give us a walkthrough on the basic must-know pharmacogenetic principles. You're an oncology pharmacist and you're familiar with using pharmacogenetics in your clinical work. But for clinicians new to pharmacogenetics, can you first explain why understanding genomic terminology is so important and how it helps in interpreting pharmacogenetic reports, for example?
Yeah, so within any scientific or clinical field, we use a lot of technical words, which forms a part of our specialized language we use to help communicate what we observe or discover. But to ensure everyone uses the terminology in the correct manner, we all need to know the basic definitions, because when we communicate with one another, for example, from a laboratory to a clinical setting, we all know exactly what is being referred to. Also, understanding terminologies, I think, is the first way in which we can demystify a complex field we enter into for the first time. And within pharmacogenetics, this is no different. We need to know the definitions we commonly use to help us understand why certain terms or phrases are used, because without this, we won't be able to interpret a report for the patient that we have in front of us.
You're right. So both you and I have a background in genomics, but for our listeners, could you break down some of the fundamental concepts for us that are important to know for your daily work, like what is genomics, what are single nucleotide polymorphisms, and why they're crucial for understanding, say, an individual's response to medications?
Yeah, so let's start with the word genomics and break it down right down to single nucleotide polymorphisms, which we abbreviate to SNPs when we pronounce it. So genomics is a study of genetic material. And this genetic material is in the form of DNA, which is a double helix made up of nucleotides. So each nucleotide contains one nucleotide base, a phosphate molecule, and a sugar molecule called deoxyribose. The DNA nucleotide bases are adenine, thymine, cytosine, and guanine. So when we sequence DNA, we want to know the order of these bases, and this order is different for each individual. We are mostly interested in the exons, which code for amino acids to produce proteins. Introns, on the other hand, are non-coding regions, which break up the exons within the gene. So when we compare one individual to another, there are up to 14 million differences, and these differences are known as single nucleotide polymorphisms, or SNPs. And it's these SNPs in pharmacogenetics that we are interested in, because it will help us identify which individuals will respond well to a medication with minimal side effects, and those who may be at risk of severe adverse drug reactions, some of which can lead to hospitalization or, in severe cases, death.
Great. So we have DNA, and DNA is made up of nucleotide bases, and then those are grouped into areas altogether, either called exons or introns, and it's those introns that help us make the proteins, and they're the things that you often look at when you're looking at a DNA sequence. So thanks, Dharmisha. I see we're building in information here, as I've just done there, but are there any other genomic terms that we should know, and the process of how genetic variation that you described there, like SNPs, influence an individual's responses to medication?
Yeah, absolutely. So let's begin with the first terminology, which is vital to know, and this is the gene. So a gene is a unit of inheritance, and it's passed from each parent to a child. And this gene contains information on how to build a protein. In humans, we have approximately 20,000 protein-coding genes, which represent around 1.5% of our entire human genome. Another word we need to become familiar with is allele. An allele is a specific version of a gene, and each person inherits 2 alleles of a gene, one from each parent. So if a person has two different alleles, then the individual will be heterozygous and homozygous if the alleles are the same. The alleles identified are compared to a reference genome, and if there is a difference, scientists will use the term variant, which means the site where the part of the gene we are interested in looks different to that of the reference genome. So now we know what a gene and an allele is and how the term variant will be used, we can now move to the term genotype, which is used throughout pharmacogenetic reports. So a genotype is defined as a set of alleles which have been inherited and will determine a given characteristic and will determine what will be expressed. Within pharmacogenetics, the genotype will indicate how much enzyme will be expressed, and this enzyme expression is known as a phenotype. And a phenotype is the observable trait. So within pharmacogenetic guidelines and in some reports, there are a couple of other words which are used by laboratories. We also need to be familiar with these, and these are haplotype and diplotype. So a haplotype refers to a combination or patterns in which DNA alleles can be found in individuals. So a haplotype can be a single gene allele or a group of alleles which can be found together. But in pharmacogenetics, we are also interested in the diplotype, which is a combination of two haplotypes, because this will determine the overall phenotype within pharmacogenetics. Within pharmacogenetics, alleles are named using a star nomenclature system, and this was developed in the 1990s, and it was used to simplify and standardize how we report the alleles within our pharmacogenetic reports.
There's a lot of information there, Dharmisha. We've gone from phenotype, genotype, variant, alleles, haplotype, diplotype. So I'd recommend to any of our listeners to potentially rewind 1 minute and then review those, and that will help kind of embed some of those terminologies. That's the beauty of podcasts. So shall we attempt to bring all of this information in together in a more practical way? Is there a clinical case study that you could describe that highlights the clinical impact of pharmacogenetics?
Yeah, absolutely. And I think it's a great idea to put it together in a case study. So let's use a case on how we can use all these terminologies and use a clinical scenario on a stroke unit. So for this case, I have a patient, Mrs. Joshi, and she has been discharged from a stroke ward 2 weeks ago. And on the ward, she had a pharmacogenetic testing done for an enzyme CYP2C19. So the enzyme CYP2C19 is encoded by the gene CYP2C19. But within literature, when you refer to this gene, it will be in italics, so you'll know the difference. We're talking about a gene or a protein. So as part of a stroke treatment, Mrs. Joshi was prescribed clopidogrel, which is a prodrug, and it needs to be metabolized by CYP2C19 to its active metabolite in order for it to be an effective treatment. There are other enzymes involved in clopidogrel metabolism, but CYP2C19 is the most important one we need to consider for this case. The results from the genomic lab are back and ready in time for her follow-up appointment. So the clinician, which can be a doctor, a pharmacist, or a nurse, they read the report. And the report states that Mrs. Joshi is homozygous for a CYP2C19 variant called C.681, where guanine has been substituted for adenine. So this means that in the exon, depicted in this case as C dot, this indicates to me that this is the coding region of the CYP2C19 gene. And after the C dot, it tells me that the reference nucleotide guanine has been substituted to adenine at the position 681. But because we use a star nomenclature system, this variant C.681, guanine substituted to adenine, is also known as star 2. And this star 2 allele represents a loss of enzyme function. So as Mrs. Joshi is homozygous for star 2, it means that her diplotype consists of 2 star 2 alleles. So therefore, what's happened here is that Mrs. Joshi has inherited 2 loss of function alleles, and this is known as her genotype. But it means that she's a poor metabolizer. And the definition of a poor metabolizer is her phenotype, and this relates to her enzyme expression, her observable trait. So when using a shared care guideline, the clinician identifies that Mrs. Joshi is a poor metabolizer and changes her clopidogrel prescription to another antiplatelet drug. This is because Mrs. Joshi cannot metabolize the prodrug clopidogrel into its active metabolite, and therefore she'll be at greater risk of another stroke if the prescription was not changed. So for this particular case, the clinician identified Mrs. Joshi as a poor metabolizer, but individuals based on their pharmacogenetic report can also be identified as normal metabolizers. And this would mean that a person has inherited 2 alleles within the gene which indicate no changes when compared to the reference DNA. And that means that they can prescribe standard doses. Additionally, you can have ultra-rapid metabolizers where an individual have 2 increased function alleles, so they're metabolizing the drug very quickly, or there can be intermediate metabolizers, and this can be a combination of at least 1 decreased enzyme function or 1 increased function allele, or 1 normal function allele, for example.
Great case study. So just, Dharmisha, just to kind of rest people assured that on a report, when you receive a pharmacogenetics report, this kind of explanation would actually be stated on there, what the genotype is, what the meaning of that is, and what that could mean for the phenotype, and then the dosing of the drug that the prescriber may be making.
Absolutely. So you'll see that you'll have the gene in its star nomenclature system and in the what I call the C dot system as well. So you'll have and it will explain that the patient is either an ultra-rapid metabolizer, normal, intermediate, or a poor metabolizer. And then you would use that information using a guideline to identify what you need to do with that prescription.
Thank you. So after all of that information, so as you know, we like to end our episodes on what's next for genomics in this space. So Dharmisha, how important is it for future pharmacogenetic tests to be designed with population diversity in mind? You mentioned Mrs. Joshi there, and you and I, for example, have different ethnic backgrounds. So what impact would that have on personalised medicine?
Yep, so this is a really important question. So in the example of Mrs. Joshi, her medical records state that she's of South Asian ancestry, and the variant identified STAR2 is more commonly found in Central, South and East Asian populations. In contrast, there is another allele, CYP2C19 STAR17, and it represents increased enzyme function, and this is more prevalent in Afro-Caribbean and Caucasian populations. So if we excluded STAR2 or STAR17 alleles on our panel, we would be omitting a huge patient populations. So it's really important to consider that our populations are also becoming more admixed as well. So when we test for genes such as CYP2C19, it really must be broad enough to reflect the diverse patient population that we serve. So a lot of our pharmacogenetic data is predominantly based on Caucasian populations, including our reference genome. So there may be many alleles that we don't know about, or we don't have enough evidence to support for inclusion to a larger pharmacogenetic testing panel. So really there is a lot more work to be done within this field in terms of like next steps. So some of this work is underway. So for example, diversifying the reference genome, but there's a lot of research being done in other countries using more diverse populations regarding pharmacogenetic research.
I can see how it's really important to include people from different populations and different ethnicity in research to be able to then prescribe in a more optimal way for people within genomic medicine. Thank you, Dharmisha. How you focused on the basics of genomics and the must-knows for prescribers in today's episode is really helpful, and that case study of a real kind of life situation that could happen and walking us through that. So hopefully we've given our listeners a good starting place to go on to learn more about anything that we've covered in today's episode.
Yeah, I really hope it provides a good introduction to pharmacogenetic terminology, and just to show how understand just a few basic terminologies can really help us navigate and interpret a pharmacogenetic report.
So thank you again. Thank you for listening to this episode of Genomics Now, a series of conversations hosted by healthcare professionals. The North Thames Genomic Medicine Service aims to complement other genomic-focused educational tools with these bite-sized podcasts. So do visit our website for more resources and updates on genomic-based clinical developments and research. If you've enjoyed this episode, don't forget to share your thoughts and spread the word by tagging the North Thames Genomic Medicine Service on social media.